Now we'll be talking about question number 5, and the question number 5 is asking, cut a transverse section of young stem of a plant from your school garden and observe it under the microscope. How would you ascertain whether it is a monocot stem or it is a dicot stem? And we have to give reasons. So we will have to take a sample of stem and we have to identify whether the sample of stem we have taken is a dicot stem or a monocot stem. So obviously any sample can be identified on the basis of some characteristics. Alright, so if we are talking about the stem, if we talk about the stem of any plant as a sample, we'll take the sample and identify the sample of stem whether it is a dicot stem or monocot stem, depending upon or on the basis of characteristics we know about the dicot stem and monocot stem. So first of all, I'm going to tell you about characteristics of dicot stem, right? On the basis of these characteristics only, we will identify the stem sample is actually dicot or monocot in nature. So here we have the characteristics of dicot stem. First of all, the dicot stems, if we talk about the dicot stems, they have vascular bundles, alright. They have vascular bundles which are arranged in a ring manner. First of all, the vascular bundles are arranged in a ring manner around centrally located pith, right? The next, if we talk about, then the vascular bundles in dicot stems are conjoint, they are collateral and they are open. That means you will find secondary growth in dicot stems which is not found in monocot stem. Alright, this is also a characteristic of dicot stem. What I've said that in dicot stems the vascular bundles are conjoint, collateral and open. That means they can have secondary growth in them. Also they have a strip, centrally located cambium strip which is present in between xylem and phloem, right? The second characteristic I have told about is the vascular bundles in dicot stems are present in a ring manner, in a ring arrangement around centrally located pith. The third characteristic of dicot stem is they have ground tissue. Yes, they have ground tissue. The ground tissue of dicot stem is differentiated into collenchyma, parenchyma, endodermis, and endodermis, pericycle, pith, etc. Right? So all these are the characteristics of dicot stem. Now, if we talk about the characteristics of monocot stem, what are the characteristics? The characteristic of monocot stem are as follows. There will be present an extra layer known as hypodermis. Hypodermis you will find in monocot stem only. This hypodermis layer of monocot stem is made up of sclerenchyma cells, right? Then if we talk about vascular bundles in monocot stem, then vascular bundle in monocot stems are conjoint. They are actually closed. That means there is no secondary growth is present in monocot stems. Also, if we talk about the arrangement of vascular bundles in monocot stem, they are arranged in a scattered manner. They are not arranged in a fixed pattern just like dicot stem. They are present in a scattered manner. Right? All these are the characteristics by which we can clearly identify that whether the sample we have taken of a stem is dicot stem or monocot stem. So let's write answer for this question. Answer 5. Okay. Answer 5 will be like this. The unknown sample of stem can be identified on the basis of characteristics of monocot and dicot stem. For this, or to identify the sample, unknown sample, we have to know the characteristics of dicot stem as well as the characteristics of monocot stem. So we will write the characteristics of monocot and dicot stem over here. In the left column, we are going to write the characteristics of dicot stem, and in the right column, we are going to write the characteristics of monocot stem. So let's write the first characteristic, that is the vascular bundles. Vascular bundles of dicots are conjoint, collateral and open. That means they can have secondary growth along with the cambium strip. Cambium strip present in between xylem and phloem. Xylem and phloem. Then the second characteristic of dicot stem is vascular bundles are in ring manner. They are arranged in a ring manner around pith, around centrally located pith, right? And the ground tissue in dicot stem is differentiated. Ground tissue is differentiated into collenchyma, parenchyma, pericycle, pith, etc. Right? These are the characteristics. These are some characteristics of dicot roots, on the basis of which we can identify whether the sample we have taken is a dicot stem or not. Right? We are talking about stem, right? Then if we talk about the characteristics of monocot stem, then you will be finding a hypodermis layer, which is made up of sclerenchyma. Then second of, if we talk about, they have vascular bundles. Yes, they have vascular bundles which are conjoined and closed and scattered. So they have vascular bundles which are conjoined and closed. That means there is no secondary growth is found in the monocot stems, and the vascular bundles are scattered. Vascular bundles are scattered, right? So these are the characteristics, some characteristics of monocot stems. We are discussing the characteristics of monocot and dicot stems, on the basis of which we can identify the unknown sample, whether the sample is monocot stem or a dicot stem, right? So with this we have completed the solution of question number